Bacteria are known to frequently participate in horizontal gene transfer. As a result, genes related by a process are often clustered in the genome, which enables them to more easily be transferred as a functional set. We can also use synthesis to intentionally move a set of related genes. One general question is, how often do such transfer experiments work and where do they go awry? In this study by Nickerson and co-workers, they examined the transfer of a type 3 secretion cluster called SPY2 from Salmonella to other gram-negative bacteria. When a mammal is infected with pathogenic strains of Salmonella, such as Salmonella typhimurium, the bacterium begins by invasion of M cells in the intestinal epithelium. This process involves a type 3 secretion system called SPY1. As a result of invasion, the bacterium resides inside a phagocytic vacuole. Salmonella has a second type 3 secretion system called SPY2, which secretes effector proteins that manipulate vesicular trafficking and thereby enhance intracellular survival of the bacteria. This process is often referred to as modifying the vacuole to prevent it from fusing with lysosomes. Like all secretion systems, type 3 secretion systems have been considered for biotech apps, including use as live vaccines, for protein production, therapeutic bacteria, and for manipulating the behavior of macrophages. In this study, the authors put the SPY2 locus on a conjugated plasmid and show that it functions in E. coli. In another similar paper, they do the same experiment but with SPY1 instead, and they show that this one does not work in E. coli. To show that it works, they monitor the expression of a protein called SSEB, which is a late expressed chaperone needed to recruit proteins to the needle complex. They also show that effector secretion happens using a similar blot assay. They employ two different media compositions that are known to cause Salmonella to either specifically express or not express SPY2. These are called MGM8 and MGM10. They successfully observe SSEB production in MG1655 and top 10, which is a modern lab strain. Surprisingly, they get different induction behavior in MG1655 than is observed in Salmonella, and that is a common reframe in such experiments. If something doesn't work in a transfer experiment between two highly related organisms, most likely it is the result of subtle differences in regulation between the two strains. Another question is whether SPY2 is sufficient for some of the additional functions observed during Salmonella infection. SPY2 was not sufficient to allow survival within macrophages. This could mean that additional gene functions are required for this phenotype, or it could be an artifact of regulatory differences. We saw in a previous chapter how biosynthetic pathways can be moved from one organism to another and how replacing regulation often facilitates such transfer experiments. This theme carries over to these other domains of biochemistry, and these complex microbiological processes often require some degree of refactoring to work.